We're going to, again, curves that Salim just told us about. This is the application of something like Moore's law applied to the cost on the y-axis of genome sequencing. And again, this is a logarithmic curve, so a straight line actually is an exponential. And what you can see is from the time we were able to read the genome, we were on an exponential curve, and then suddenly in 2008, the cost of sequencing the human genome started outpacing Moore's law, and it dropped very, very rapidly. That's when next-gen sequencing came along. Now we can not only read the genome, but we can write to the genome. We can manipulate the genome using something called CRISPR. And this is actually what CRISPR looks like. It's sort of an animation of it. But the fundamental concept is that we can take enzymes, proteins again, and we can use them to scan through an organism's DNA and look for misspellings and then correct them. So it's like the Control F, Control X function of Word or a processor. And we can do some very interesting things with that. So one is we can actually create new living medicines. It turns out bacteria can be infected by viruses just like we can. You can actually see an example in a cartoon to the right of your image, where you see a viral genome being injected into a bacterial cell, which gets it sick. But we can now package the kinds of things we want to kill bacterial cells. And this is a way of making medicine, because we can kill bacteria now that have become resistant to our antibiotics. Another thing that we can do with CRISPR by rewriting the genetic code is to repair base misspellings that cause human disease states. So the very first FDA approval to treating human disease happened in 2023. The FDA approved basically using gene therapy and CRISPR-Cas9 to treat sickle cell disease. Now, there are other they seem kind of out there, but they might not be.